From the top next, please. Okay. So hello and good afternoon, everyone. So yes, we are the first group to present with our study entitled Antibacterial Activity of Formulated Soap from Citrus aurantifolia, which is known as key lime, and Pisidium guava, also known as guava, leaves ethanolic extract against Escherichia coli. So allow us first to dig upon with our introduction. Next, please. So basically, in this study, we are going to investigate the antibacterial activity of both or joint plant materials, which are the Citrus aurantifolia and Pisidium guava ethanolic leaves extract, together with the formulated liquid soap of which. So for the aspect why we choose these given plant materials is that we consider first these plants that they have already been proven scientifically by its antimicrobial activities. So why key lime? Why guava? So given the fact that they are already been proven by their antimicrobial activities, so um, we would like to investigate further if these plants would totally kill, would still totally kill a certain kind of bacteria, but in a fused manner. So they are proven individually, separately. So in this study, we are going to fuse them. We are going to fuse the both plants, their ethanolic extract of the both plants, and as well as we are going to formulate a liquid soap from which plants. Um, and for the aspect why we stick on only with its antibacterial activity is that we would like to be more specific, considering also the fact that we will only be focusing on a certain type of bacteria, which is this Escherichia coli. So yes, we choose ethanol as our solvent, ethanolic extract. Ethanol is our solvent. For basically, ethanol has been um, known as a good solvent for polyphenol extraction, and it is proven by it is stated by the Food and Drug Administration that it is effective, efficient, and at the same time, it is safe for human consumption. And um, and at the same time, why among all the bacteria, why E. coli? There are lots of bacteria, why E. coli? So we highly put an emphasis with this bacteria, namely this Escherichia coli, considering also the given factual information and statistical information rather that is included in our paper about specifically about how massive it affects yearly in terms with its bearing in bearing bacterial infections. And as per stated by the World Health Organization in the year 2017, there are at least 600 million individuals get infected by this certain type of bacteria. And among those 600 million individuals, 32,000 of which belongs in the Philippines alone in view of water and foodborne diseases. So why liquid soap, and for the aspect why we choose a vehicle, why our vehicle is liquid soap or why our medium is liquid soap, is that cliche may it sound, but as we all know, uh, hand washing is the single most effective way to prevent first the spread of infection. And in terms of the existing synthetic liquid soaps that we have now, most of those products kasi um, involves this certain type of chemical which we called as triclosan. So basically, this triclosan is the most frequent antibacterial ingredient found in the existing antibacterial or antimicrobial soaps that we have now. So in a ratio of four liquid hand soap, three of which contains triclosan. And why is it? I did mention about this triclosan. There are evidences that we've stated also in our paper that this triclosan can result into bacteria strains that are resistant in the long run for disinfectant and antibiotics. So this basically means that um, uh, by washing or by using synthetic antibacterial soap to wash your hands, you're just merely exposing yourself and the environments to higher levels of these chemicals with no discernible benefit in the long run. Not just in this specific period of time, but in the long run. So, yes, basically, we would like to formulate a certain antibacterial liquid soap that comes naturally with the use of these plant materials that we have. So next, please. Yes, Princess. Ses, hindi ka namin marinig, Ses. So for the statement of the problem, our first statement of the problem is that what is the minimum inhibitory concentration of the Citrus aurantifolia, which is the key lime, and the Pisidium guava leaves ethanolic extract against Escherichia coli? And secondly is that what is the mean zone of inhibition of the following treatments against Escherichia coli? So this involves the combined Citrus aurantifolia and Syzygium guava ethanolic extracts. And at the same time, we do have this positive control with specifically this ceftriaxone. And for the negative control, we have this distilled water.
Hello, marinig na ako?
Yes, yes.
Sige. Next text. Okay. Number 3, what is the mean zone of inhibition of the following treatments? Again, Escherichia coli, Citrus aurantiferia, key lime, and Pisidium guava leaves ethanolic extract formulated soap, um, ceftriaxone, positive control, distilled water is the negative control. Number 4, is there a significant difference between the mean zones of inhibition of Citrus aurantifolia, key lime leaves ethanolic extract, Pisidium guava leaves ethanolic extract, ceftriaxone, the positive control, and distilled water, the negative control against the Escherichia coli. Next, Dex. Number 5, is there a significant difference between the mean zone of inhibition of the formulated soap from Citrus aurantiferia, key lime, and Pisidium guava leaves extracts, the ceftriaxone positive control, and the distilled water as the negative control against Escherichia coli? Number 6, is there a significant difference between the mean zone of inhibition of the Citrus aurantiferia, key lime, leaves extract, Pisidium guava leaves extract, and the formulated soap from the two plants against Escherichia coli?
Next, please.
Null hypothesis, number 1, there is no significant difference between the mean zone of inhibition of Citrus aurantiferia, key lime leaves ethanolic extract, Pisidium guava leaves ethanolic extract, ceftriaxone, the positive control, and distilled water as negative control against Escherichia coli. Number 2, there is no significant difference between the mean zone of inhibition of the formulated soap from Citrus aurantiferia, key lime, and Pisidium guava leaves extract, ceftriaxone as positive control, and distilled water as negative control against Escherichia coli. Next, number 3, there is no significant difference between the mean zone of inhibition, the Citrus aurantiferia, key lime leaves extract, Pisidium guava leaves extract, and the formulated soap from the two plants against Escherichia coli. Next, Dex.
For the significance of the study, first, it will benefit the community to offer an alternative method to reduce Escherichia cases to those who are susceptible and exposed with this bacteria. Next, for the Department of Health, uh, this will help them to recognize that this study will be beneficial to the community in promoting natural and profitable treatments for the Escherichia coli. Also, this will provide the Food and Drug Administration or FDA another source for liquid soap with an antibacterial capabilities. Next is for the pharmacy students for them to apply and test their classroom information to their experiment as well as appreciate the significance of their pharmaceutical education. And next is for the academe, for the institution, uh, it gives knowledge and learnings to the individuals or to the students inside the school regarding its essential use. Next is the researchers to have an adept understanding in regards with this study. And lastly, for the future researchers I, conducting study on this topic will use the findings as a guideline or reference material. Next, next na Dex. So for the conceptual framework, the independent variable are the ones being manipulated or investigated in the study, which are the citrus aurantifolia or key lime and Psidium guava leaves ethanolic extract, formulated liquid soap from the citrus aurantifolia or key lime and Psidium guava or guava leaves extract, and cetriaxone as the positive control and distilled water. And the dependent variable of the study is the antibacterial activity of the two plants, which will be measured by its zone of inhibition. Next.
So for the methodology for the research design, so this study is a quantitative through experimental one. Specifically, will utilize a post-test only control group. And basically, the research local will be then this research will be conducted then in General Santos Doctors Medical School Foundation Incorporated, specifically at Pharmacy Laboratory. And for our sampling technique, we do have purposive sampling for the plant materials that we have, since we have set a criteria for choosing our plant materials. Basically, specifically, we will choose a leaves that is devoid from flaws, holes, and bugs. That's why it is purposive sampling, and random sampling for the Petri plates of procured bacteria. And basically, random sampling will be applied for they every Petri plates will be randomly assigned into groups. Next, please. So data garnering first procedure first, we will seek first a permission from the um, respected individuals that are involved that are needed for this study. Secondly, is that the preparation of the plans. There it is included there the collection, the authentication, and instruction extraction of the plant materials. Collection, in terms of collection, we choose to collect our plant materials basically in specifically ang specific time is that in a morning warm sunny day, morning and sunny day. And then basically we will be then seek seek to we will seek a botanist in order to authenticate our plant material itself if it is suitable and accurate one. And then extraction itself, the extraction of the plant materials, we will then wash first the plant materials. So I separate first the guava and the key lime, wash them, and then dry the leaves, and then we will be submerging it in a phenolic extract, and then be cut into pieces, and will then go into a rotary rotary evaporators as cool, and then right after that we will filter it first right after before we will do it in a rotary evaporation of the pharmacy laboratory na given sa laboratory itself. For the preparation of bacteria,
For the preparation of bacteria, once the researchers will first procure the cultured bacteria in NDDU. Then once it is procured, the researchers will store this in the lab refrigerator in 4 degrees Celsius to maintain or to make sure that the bacteria will not desiccate. Next would be the preparation of dehydrated Mueller-Hinton agar. So this would be first, the researchers will first make sure that their area of workplace is sanitized, the equipment is sanitized, and they must wear a PPE. Then they will measure the proper amounts of the ingredients, heat these ingredients, then undergo an autoclave for the final sterilization. Then after that, they will make sure that they will cool the prepared Mueller-Hinton agar at room temperature. Next, please. Next would be minimum inhibitory concentration, or specifically, this is a dilution method. We'll be focusing more on broth medium. So the researchers will, of course, again sanitize their areas, equipments, and of course wear PPE. They will prepare the Mueller-Hinton broth, then prepare the necessary test compounds, specifically uh, the leaf extracts of the key lime and guava. Then after that, they will prepare to conduct the test. They will prepare that sterile test tubes and add the Mueller-Hinton broth. Next would be the test compounds. Of course, leaving a test tube for as a positive control. Next would be the E. coli in these test tubes. Then incubate these test tubes for at least for at least 1 day. And after that, the researchers will interpret or observe the results after the 24 hours. Next would be the distribution of media. Specifically, this media would be the agar Mueller-Hinton agar. So this is just a continuation of preparation of Mueller-Hinton agar before. So after the agar has cooled down in room temperature, they will use an aseptic technique, and then they will pour the agar in a petri dish, making sure that it is in a 4 millimeter depth. And depending on the size of the petri dish, the amount of agar poured in will be different. Next, after that, is that they will be stored in a refrigerator, or specifically the laboratory refrigerator. Next is the preparation of this diffusion method. So the researchers will prepare the filtered disc, then of course the tested or the test compounds. Next would be they will inoculate the E. coli in the prepared agar medium, and then making sure that their lab equipments are of course sterilized. Next is the formulation of liquid soap.
For the formulation of liquid soap, the hot process method of Debnat et al. 2011 will be utilized. So first, To make the lye solution, we need to combine the potassium hydroxide with the distilled water. Then the heated glycerine will be placed in a beaker. In a separate beaker, the coconut oil will be heated. Then the lye water will be then added after the heated oil will reach 60 degrees Celsius, followed by the heated glycerine. After that, the solution will be continuously stirred until it forms a thickened substance, and that will be the soap paste. Next.
So statistical analysis, this will be used in analyzing and interpreting the collected data. Number one, the mean. This statistical tools is used in the study in order to compute the zone of inhibition of the citrus aurantifolia and the Pisidium guajava. Number two is the standard deviation. This statistical use is used in the study for the researchers to determine the degree of variation of the both groups. Next is the analysis of variance, ANOVA. This statistical tool is used to determine if there is a significant difference between the mean zone inhibition of the citrus aurantifolia and Pisidium guajava ethanolic extract against the Escherichia coli. And the post and last is the post hoc least significant difference. This statistical tool is applied in order to determine which among the treatment groups have a significant difference between them. But if the result in an analysis of variance is there is no significant, then the post hoc least difference will not be included. Next po. Next.
Ethical consideration. First, let us talk about approval. In conducting an experiment, it is very important to confirm approval from the instructor. It is a must and it is considered in doing research as an ethical one. Conducting conducting of experiment, in order for us to sum up with a result, the first thing that we will going to do is to conduct an experiment to conclude an exact result for our study. Proper handling of bacteria. In doing our experiment, it is very important to be careful in handling bacteria for our safety purposes and also for the accuracy of our results. We should wear proper PPE. Last is the proper disposal of bacteria. It is very important to avoid biological hazards. It is ethical to be responsible in disposing it properly.
In conducting the experiment, moreover, in conducting the experiment, the researchers are supervised by their research advisor, particularly in handling bacteria in order to avoid any unnecessary things. Next is the proper disposal of bacteria. The apparatuses or the materials used in this experiment will be sterilized and placed in autoclave bag for 30 to 40 minutes. And those materials or apparatuses that has no contact with the bacteria will be sanitized for with the 10% bleach before putting them in the respective area. And proper handling of bacteria, of course, the researchers are told to must wear their proper PPE equipment and they must sanitize their hand before and after conducting the experiment, as well as the area must be disinfected before using. That's all.
Okay, thank you so much. I think they are done, Ma'am Reza. Okay, can we ask all the proponents to kindly unshare your screen so that we can start with the Q&A? I would like to start asking Mr. Dex Espanol. You have written in your manuscript, no, that you will be using a true experimental design, specifically the post-test design. Can you describe what is your understanding about post-test design? Why is it you call that a post-test design? Ano yung gagawin ninyo? Na? Because I have read in your manuscript that you will be using the post-test design. So meaning there is no pre-test, kasi post-test na. Anong gagawin niyo doon? Because you'll be using, of course, you have also your control and experimental. Will you please describe using that design? Although it was presented, no, to the panel about how you're going to do, no, with your sample, your extracts. Mr. Espanol, I want to hear from you. Naka-mute ka. Okay. How about Miss Pipito? Miss Pipito? Can you hear me? Am I audible?
Yes, ma'am. Yes, ma'am.
Yes, ma'am. So we will use true experimental, quantitative true experimental post-test only control group because we will only measure the zone of inhibition after the intervention.
Kindly unmute.
Hindi po ako narinig, ma'am. Wait.
Narinig ka namin, naputol ka kasi nag-mute ka kaagad.
Ay, sorry po. So, we will use true experimental because we will examine the contrasting groups which the other one will be exposed to the intervention and that will be referred as the treatment group, and the other one will not be exposed to the intervention of that will be used in the study, and that will be referred as the control group, ma'am. And then also we will only use post test only control group because we will only measure the zone of inhibition after the intervention, ma'am.
Okay, thank you. Mr. Espanol, I want to hear again from you. It was mentioned several times about purposive sampling. Why is it purposive sampling? Mr. Espanol?
Go Dex, kaya mo yan.
Akala niyo, Mr. Espanol, no? Kasi this will be your study, and I'm asking because it was mentioned several times about purposive sampling, sampling technique. So why is purposive sampling? How it is being observed in your study?
We set we set some criteria. We set some criteria on plant materials, ma'am.
Okay, specifically, what criteria did you consider in the selection of your material?
Young plants, young leaves, ma'am.
Okay, so you'll be using young leaves, and aside it's very young. How would you describe that yung young? Kagaya sa amin, young man kami, young at heart. So, paano mo masabi na young din yung leaves na yan? In terms of days, months?
Ma'am, no holes and flaws bugs.
Okay. Pero yung young, how would you describe specifically? How would you, paano masabi na young leaves na siya? Do you have specification in terms of the number of days from the start na naglumabas yung dahon, ilang days siya para masabi mong young? No? Kagaya sa tao din yan, kagaya si Ma'am Gabilagon, very young. Kami ni Ma'am Eroy, young lang. Pero si Ma'am Gabilagon, very young.
Young at heart.
Young at heart. Yeah, Mrs. Espanyol. Meron kayo yan, Miss Gatan?
Yeah, we consider the leaves of the plant na young, ma'am. It is basically the either side of the stem that is considered as the young ones. And then at the same time, they have these short petioles. These short petioles of the leaves, that would be the hint that it is a young one. And at the same time, yung stalk niya, ma'am, yung stalk ng stem niya is a bit, hindi pa siya yung firm talaga. Unlike with the grown one or yung mga mature one na leaves, ma'am.
Iba talaga ang young, no? So kahit din sa guava leaves, yung dapat young talaga. Okay, Ma'am Ray, sa mga young na criteria for posted communication. Okay, so thank you. I think Ma'am Eroy and Ma'am Pinko and Ma'am Raysa, if they have some questions, later na ako magbigay ng aking mga suggestions. Baka may mga initial questions po sila.
Ma'am Queen ko, ikaw una, ma'am.
Ikaw na, Ma'am Eroy. Do the honor.
Okay, based on when I have read your paper, no, I would just like to ask if you have said a while ago na you will be testing the antibacterial activity of the formulated liquid soap, at the same time test the antibacterial activity of the ethanolic extract. Am I correct in my understanding of your paper?
Yes, ma'am. Yes, ma'am.
Okay, so my question is, why combine the two plants? You have said a while ago that there are plenty of studies that talks about antibacterial activity of the guava, at the same time antibacterial activity of your key lime. So why is there a need to combine the two plants?
Basically, ma'am, we just wanted to investigate whether if the combination of the both plants will exhibit a stronger upshot or not against a certain bacteria, which is the Escherichia coli, ma'am. We would like to put an highlight for the stronger upshot, since for the individual studies and evidences by these plants, antibacterial, antimicrobial activity nila is that they do have this inhibition, but moderate ones only. They have this moderate antimicrobial activities against certain types of bacteria, ma'am. We also included in our research paper.
But which among the two plants has the highest antibacterial activity?
Citrus aurantifolia, ma'am.
Okay, so if you have said that if citrus has the highest antibacterial activity, why not focus more on citrus? And then let's say, for example, now enhance more of the studies. If you look at the previous batches we had with from the previous students, we have plenty every semester we have always antibacterial studies, no. There are those studies na the same plants but other parts lang are being investigated. One that I could remember na medyo kakaiba lang, if you talk about antibacterial, I don't know if Ma'am Risa would agree, is with the nanoparticle katuganing kay Elise yata to raise. So if you are to compare the previous antibacterial study of the pharmacy department, almost the same lang. So ang question ko is, why not let's focus, this is more of a recommendation ha, kay taas kaayo inyong trabaho uno, right? You are to investigate the formulated liquid soap and then check again the antibac of the ethanolic extract. Why not exclude the ethanolic extract? Because why not exclude the ethanolic extract and then you can add something like, let's say for example, if you want to focus on the formulation of the soap, halimbawa, pwede ninyo butangan o let's say for example, antioxidant activity ba niya, or let's say deodorizing effect ba niya. If you have identified na kana nga plant, si citrus, has the highest antibacterial activity, so ato na lang i-enhance more si citrus, bringing the presentation of getting the highest antibacterial activity.
Ma'am, for the existing proven scientific na studies by the citrus or antiphilia, more likely and most study kasi ma'am, nagadeal lang sa citrus or antiphilia fruit itself, not by its leaves. So here in our study, we would like to utilize the plant leaves itself, ma'am. And yes, basically, leaves only, ma'am.
So may mga studies na fruits ang mas mataas ang antibacterial activity.
Yes, ma'am.
And then kayo ibahin nyo lang, so you would also want to know the concentration of antibacterial activity doon sa leaves ng citrus lime. So is there a need to add another plant, to combine it with another plant? Nakamute ka, Pauline.
Considering the fact, ma'am, that guava, I've included or I've combined certain plants. I've combined these two plant materials because I did consider the fact that this plant guava, Pisidium guava, is highly recognized as one of the listed and recommended or approved by the Department of Health as one of the medicinal plants. So the reliability bitaw, ma'am, and at the same time, more the studies that exist din, yung mga existing na studies about this guava, more likely nagadeal din sa fruits niya. So there are several fruits and leaves niya itself. So I've come and pondered to the point na why not combine yung dalawang leaves if they exhibit a strong inhibition in a certain bacteria, ma'am, para mas stronger yung concentration ba or what?
Miss Pauline, in addition to what has been raised now by Ma'am Eroy, because she is looking into the process, and later on, if you'll be studying both no guava leaves and lime, it will take yung time Ma'am Eroy no ng pag-study nila sa yung process. And at the same time, you made mention also, it is well studied already. Even the Department of Health, you made mention na talagang it is being recommended, no, because of its antibacterial effect. At saka parang madami na rin Ma'am Eroy and Ma'am Reisa sa department niyo ng mga studies sa guava leaves in the previous years.
One of the
meron na?
ang ano ko lang dyan din is the guava itself is kana very common plant, no. So would like to recommend unta if you have said a while ago that if the key lime has the highest antibacterial activity, why not focus on the key lime and then add another activity? Let's say deodorizing activity. Para sa liquid soap, ang inyuhang package jud is the liquid soap plus na ba siya deodorizing activity, or would you like to add antioxidant activity? Ana siya. Except unlike if we will add another plant nga according to you kuan siya, well studied na si guava as antibacterial.
Can I butt in, ma'am?
Sige daw, ma'am.
For me lang, ma'am, ha, kasi your concern is about the time of the study, but when in fact they
Not
are
the
just
time.
ay kay si Din.
Well studied na siya, yung guava.
Ay,
Well studied. Ingon niya well studied ang guava. So it's not about the time.
Kagaya na na-mention mo good ang time sa pag-conduct sa study. So I was thinking na since this one is this same na pharmacological action, it would be easier for them than to add another pharmacological action that is a different procedure. So mao lang to ako ang gusto ingon unta when I heard the time of the kasi they can do this at one time lang kay isa ra man siya ka activity.
Moreover, ma'am, this study basically nagaano din siya, ma'am, naga-infuse siya sa usage ng liquid. Infuse namin yung combined extracts ng both plants, infuse namin sa vehicle as a liquid soap. So I haven't seen most likely mga combined plant materials extract na infused sa liquid soap, ma'am.
Okay. I don't know, no. With the ethanolic extract, why is there need to you will test the antibac with ethanolic and then extract and then formulate it into a soap? Unsa tong imong giingon na i-infused?
Yun, ma'am. Separate yung ethanolic extract ng both plants and at the same time, separate yung formulated soap ng dalawa. Bali,
so how many products will be produced? Dalawa. Formulated soap of the guava and the formulated soap of the citrus. So you will be testing all the antibacterial activity, formulated soap of the guava, formulated soap of the citrus. Tapos,
no, ma'am, combined plants na rin yung soap namin, ma'am.
Okay, combined plants, formulated soap of guava and citrus, and then test the antibacterial activity of guava alone and citrus alone. So tatlo, you will be testing 3, making testing with MIC yung this diffusion na tatlo.
The combined plant materials ethanolic extract, yung ethanolic extract ng both plants is combined also, ma'am. And at the same time, yung formulated soap niya is combined.
Combined also?
Opo.
So dalawa?
Yes, ma'am.
If may I ask po, no, as I read your methodology, so you said na combination, but I haven't read any calculations or ratio and proportion of the combined, no, kung paano nyo gagawin, gaano karami yung ethanolic extract ng guava, gaano karami yung ethanolic extract ng lime. Tama ba ako, Gatan? Or lead me to your paper kung saan doon nakasulat. And even in the formulated liquid soap, I don't see there your no, sa method niyo, I don't see there mga naiisip na calculations, how will you produce a 1,000 ml together with your extract. Tama ba, Gatan? Wala doon. Sige. orient us, paano nyo gawin yon? Or anybody from the group? I think si Pauline lang yung sumasagot. Naka-mute ka rin, Pauline. Or baka merong gustong sumagot anybody from the group.
Miss Joy Marie Barcelete.
Yes,
And Miss
ma'am.
Almeida.
So, most of the, yung base na makita po doon sa, in the research paper, it focuses more on general pa po, general. So, kasi it's hard to collect po kasi ng extract muna ng leaves kasi most of the time, bundle po yon. And so we cannot say muna how many or how much ml or how much extract is needed or measurement pa po. Then that's why in formulation of liquid soap,
So with that, you need ilang trials hanggang maabot nyo yung yung required zone of inhibition. So you're going to start with yung baseline ninyo doon. Tama ba, Ma'am Risa? Kasi wala pa silang calculation. So saan sila magsa-start
That's
ng
the use.
formulation nila?
That's the use of the minimum inhibitory concentration. That is why the
Okay.
first SOP is asking about the minimum inhibitory
Minimum muna.
concentration. And that will be used in the formulation also.
Okay.
So they cannot actually include the concentration since they are going to do that pa in the procedure.
Kaya yun ang nakikita natin,
Ma'am?
medyo mahaba-haba nga talaga ang process kasi ang pag-combine ng dalawang extract.
Yes, ma'am.
Parang
Para gwapo ba, is it not possible to add another activity for an antibac?
What do
Na
you
kanang
mean?
medyo sayon.
What do you mean, ma'am?
Let's say, for example, antibacterial, isa lang ka plant, and then add another activity, antioxidant ba?
And that is another pharmacological activity, ma'am.
Oo, pero is it not possible na test one plant and then two pharmacological activity, testing kung gusto nilang antibac, let's say antibac and antioxidant activity or antibac and deodorizing activity, deodorizing whatever. Kay ang ilang i-formulate man is soap. So why not kanagong parehas gud sa ubang mga commercial soap we had nga with an antibac activity at the same time it has some additional pharmacologic activity, di ba? Na siya antibac,
what do
like
you call that?
fungicide, like
Yung hindi maka-dry ng skin. Ano ba yun? Ano nga yun? Yung hindi maka-dry ng skin.
moisturizing.
Moisturizing.
Just to add lang para nice ba and to make it different from other studies na antibac nga the same.
I think we will have problem with the procedure, ma'am. We have to look for the procedure first before we can decide if we are going to really add those pharmacological activity, because ever since we have not yet conducted deodorizing activity and moisturizing activities sa school. Medyo mahirap siya i-test.
Let's say if it's not feasible sa school, try to consider lang, let's say for example, ask ba Sir Bado on how to do it para lang may additional na activity ang isa ka plant.
I cannot decide. It's up to the
I think
student.
it's the students who has to
Yes,
who needs
ma'am.
to answer this question.
I cannot answer also because I don't know the procedure and how to do those things.
Try to consider lang daw, Miss Gatan ba, para nice din ang papel, mas ma-enhance more, because since the day with, since the batch with Cherry, we always have every year antibacterial activity. Naulit ang ibang
We only
plants.
have, so far, we only have 2 antibacterial activities na study sa school for the pharmacy department.
2
How
since
about
si
ano?
Ma'am Panerio's time.
Yes, ma'am. Si Elise ug si itong classmate ni Elise. Oy, duha pa na sila, ma'am, E. coli ug staph
Ever
aureus.
since.
Yes,
Kay
ma'am.
Ma'am Panerio nga time. So kinsa tong naggamit iko ay mga other
Si-
activity sa medtech?
Oh, yeah, medtech, ma'am. Sa pharmacy, we only have 2. The one that was formulated, ay reduced to nanoparticles sa Staphylococcus aureus to siya. That was the study of Elase. And then the other one is E. coli for the group of Mistenizo.
Kayo, try to consider lang it.
Ma'am, if I were to ask siguro kung mag-add kami ng another activity, siguro
Pero mag-add
maisip
kayo
ko
ng isang
ano,
activity, pero isang plant na lang. Yung sabi mo na mas mataas ang antibacterial activity, si citrus na lang.
I don't know with the activity niya ano yung reaction niya with the anti-inflammatory activity, ma'am? Sa citrus.
What do you mean anti-inflammatory?
Another activity ng citrus.
Hindi, kasi I'm thinking of your final product would be a soap. So I'm thinking of enhance the soap ba with suggestion ni Ma'am King ko moisturizing activity. Mine is deodorizing effect. Anak good. Kay your final product would be a soap man.
For the moisturizing activity, ma'am, I think it has something to do with the formulation of the soap itself lang po. So baka pwede rin siguro, ma'am, na ma-add up yung moisturizing activity ng soap.
Actually, nag-research ko mga moisturizing activity. Those are tested sa tao. Sa tao. Pero let us see.
I have read some of
in
the
vitro.
studies gani raised with deodorizing activity.
Yes,
Ang
ma'am.
ginagawa lang nila no kay color lang with acidic solution and basic solution. Check on that lang para magwapo lang ang papel. Bright man ni si Miss Gatan and the rest of the gang. Kaya nila ni para
Unsang
manice
color, ma'am? Ang moisturizing activity, ma'am, we need dili siya kaya sa school.
Ang deodorizing.
Yes, ma'am.
Deodorizing activity. I-check nyo daw ang deodorizing activity kung kaya siya. Or if not, if it is not possible sa school, mag-outsource.
Roy, may I interrupt? No, we need to consider also that there is a coming Delta variant, and I think some cities magkakaroon ng mga lockdown.
Okay, yeah, I got Dean's idea of not allowing him to go outside. Okay, Dean.
Yes, ma'am.
To go outside.
There are some restrictions now we need to consider. If there will be some restrictions, especially that there's already a Delta variant in Davao City. So if there are some restrictions in other cities, so our students, if possible, we will simplify their studies that will be accommodated only here in our institution. So I know the idea and the recommendations are very good, yet for this time, I think we need to take some consideration with the current situation po. Sorry.
Okay, Dean. So that's noted. Itong procedure niyo, Miss Gatan, sa school lang lahat i-conduct?
Yes, ma'am. Yes, ma'am. from the extraction down to the formulation of the liquid soap itself, ma'am.
Okay. So can we move? Mab. Um, anyway, those are just some of the suggestions, no? But another question that another thing that I have noticed ba with your positive control, uh, ceftriaxone. Tama ba yun, ceftriaxone?
Yes, ma'am. Ceftriaxone.
Why ceftri?
Ceftriaxone, ma'am, basically it is under the third generation of cephalosporin. So this cephalosporin, as we all know, is effective against these certain gram-positive and gram-negative bacteria. Ceftriaxone, to be specific, sa under siya sa third generation, right? And this ceftriaxone is the only drug that I've searched, ma'am, na has this greater inhibition in terms of the Escherichia coli. And considering also the fact that the phytochemicals of the both plants, specifically the limonene, same sila ng mode of mechanism of action ng phytochem at saka itong ceftriaxone, which is ang target nila both is the cell wall of the bacteria itself.
So parehas ng MOA. Si
Yes, ma'am.
ceftri.
At saka yung
yes,
phytochem na present for the both plants.
Okay, can you not consider other oral cephalosporins na with the same MOA with your plants? Di ba kay murag injectable si ceftri? Tama ba,
Yes.
ma'am?
Yes.
Pwede
I
mag-ask.
siya actually.
Pwede mag-ask, ma'am, why do we need to consider the oral preparation when in fact they are formulating soap man?
Ay soap man ilahad ay no.
Yeah.
Di man dahil nila ni ipainom sa ilaga.
Yes, ma'am.
Oh yeah, it's soap. Then go back to our recommendation. Do the moisturizing and the deodorizing activity.
Yes, ma'am. We will then add
Para check kuan siya kanang di man dahil mo mag-ilaga.
Yes.
Diba? Para pagwapa lang good Miss Gatan.
Wait lang.
Yes, ma'am.
Nabisi kong pangita o procedure sa duha.
I-check niyo.
Kung kaya ba siya himuon, ma'am?
O, kung kaya lang. Kung kaya lang siya. Himuon. Dili man dahil mo mag animal testing.
Yes, ma'am.
Okay, so I think those are just some of the questions. Now, can I pass it on to Ma'am Mabe?
I think some of my questions are already thrown, no? So, I guess proceed na lang po tayo siguro sa page by page. Diretso na lang ako doon magtatanong po.
Wala na akong question page by page map except that I understand na when I have received this manuscript, students, medyo wasak siya pero according to Ma'am Risa, minsan tama ba ikaw to Race? Magsabog ang
MS Word.
ang file sa MS Word. So I do not have so much questions with the page.
I think, Ma'am Eroy, I think there are some minor, mga minor recommendations lang, page by page, no? As what has been mentioned by Ma'am Mabe, may mga recommendations lang tayo pa konti-konti lang. It's more on mga technicalities. And I think, like for example, in the title page, wala na kayo using only one fruit with the title, ma'am. So we need to remove the guava leaves. Antibacterial activity of formulated soap from citrus. So that can be against E. coli. So tanggalin na po ba natin itong guava leaves sa title page natin. Agree na po kayo, Ma'am Eroy? Kasi based on the recommendation na has been discussed kanina, tanggalin yung isang
My concern man po din if tanggalon siya, medyo simple study na siya. For now,
as
for
is na lang
now,
nila, Ma'am, pa-combine na lang.
as is, pa-combine na lang siya if,
Sige.
but the recommendation is to enhance the soap. If they can find some procedure that can be done in school with deodorizing and moisturizing activity, they can remove the second plant na guava.
Okay. Okay, so as is na lang po muna yung ating ating title, no, in our title page. Except of course with the date, no, masyadong advanced na sila, April 2022, it's still July 2021. Naka April 2022 lagi ito sila, Ma'am, no? 2021 pa man lang tayo. Siguro advance lang talaga ang nag-type.
Kasi
Final defense
they
na
are
tayo.
project, yeah, actually gi-pattern ako na siya sa completion sa ilang study din. Kaya sa ilang scope.
You will be conducting it, di ba, by first sem.
Dili, second sem daw.
Second sem pa
Second
din.
sem na nila. So
Yes,
summer
ma'am.
na lang research 1, research 2 will be second sem.
Second sem.
So April, o but the proposal na karoon July 2021. Kaya pala nag-April 2022 sila. And also, kindly check lang the grammar no, with your acknowledgement. Mamabi, meron kayong correction. Sa akin yung spelling ng nietes, ha? N-I-E-T-S. N-I-E-T-E-S, not N-E-I-T-E-S. And also, may mga corrections na ako. I'll be forwarding that. No? Siguro yung mga statements ninyo, no? Kindly include, kindly improve that one. Mama Abe, meron kayong additional sa acknowledgements?
I would like just to comment din kung third person ba yung gamitin dito or pwede sila mag first person sa acknowledgement. Kasi ginagamit nila. Or third person tayo, the research.
Third person, ma'am?
Oo, dapat ang gamitin din. Researchers, parents, families, ganun po, researchers, advisor. Kasi R ang ginagamit nila dito. It would be formal ba din or okay lang na first person ang gamitin sa acknowledgement. Like to our parents, to our friends. Okay lang ba? Kasi we used to write the acknowledgement in the first person. Rephrase na lang. Yun lang naman ang ko din.
Okay, sa table of contents, meron ako ditong suggestion, no? Like sa appendices, kindly specify, no, here sa appendices ninyo include other letters like letter to the Research Ethics Committee, hindi ninyo nasali. Additional pa kayo sa table of contents. About with the introduction po.
Okay na ako actually sa introduction din. I think 2 pages lang din naman sila.
With the SOP.
Ito, kaya to lahat, 6 SOPs. Ang dami, no? Anyway, andyan si Ma'am Reza to help. Ma'am Rez. So you said kanina kaya naman to sa time frame, allotted time to conduct.
Yes, ma'am. Actually, pwede na nila sabay yung tanan. Ang katong ila Isobel dati, they have 8 or 9 SOP. 7?
Okay na ako din.
Okay, about significance of the study, ma'am. Aside from the community, DOH, FDA, pharmacy students, academe, researchers, and future researchers.
pharmaceutical industry.
Mhm.
Can we add on? Kasi parang more entrepreneur. Pwede kayong maging entrepreneur after this.
Oo. So ipapaan natin yung
Yes,
pharmaceutical company.
industry. Yes, the
Industry.
pharmaceutical industry.
Okay.
With the theoretical framework, so if tanggalin yung guapa, delete na lang po siguro dito if ever natatanggalin.
But based on the recommendations sa title page, ma'am, walang tanggalin. So as is na lang even the
As is na
conceptual
lang.
framework and the conceptual framework, no?
Yes.
Dito po sa study nila. In page 14. So RRL,
So RRL, I think wala silang synthesis towards the end. Can you add on synthesis ninyo for the RRL and the RRS? Yun lang yung naano ko dito napansin.
ma'am, for the research design, do you have any additional recommendation on how they stated and described their design?
We have to mention pa ba dito true experimental analysis. So when you say true, meron ba untrue or false? Or kung ano na siya, general experimental analysis na lang, quantitative experimental analysis. That's my own ano lang dito. Para medyo hindi na malito sa reader or sa anybody na magbasa. When you say experimental, quantitative experimental research design, okay naman siguro yun.
Actually, ma'am, sa kanila is naka-specify talaga siya kasi minsan we have so many experimental designs. So sa kanila kasi wala namang pre-test na gagawin. So automatic, they will be preparing the soap using the different extracts. And then after that, they will have the post-test with the SOP. Actually, sa akin, okay na itong kanilang post-test design. Looking into the SOP written by the group. Unless, Ma'am Eron, may additional pa siya. Doon sa research design, Ma'am.
Sorry din. Sige, mag-upload mga si Sir Harvey. Maraming enroll sa pharma. Kamagan din. Sorry.
Okay, may additional ka pa doon, Ma'am, sa research design. Para sa akin kasi okay na. Consistent naman siya doon sa mga SOPs. Makita naman natin that they will just have the intervention using the soap, no? And then after that, it process nila, titingnan nila yung result.
Okay, so sa research design, quantitative, mention naman through experiment, na-mention na ano pa, experimental.
So tayo na dito, no? Even with our research local.
May I ask from the group, sinong taga-barangay Conel? So si Tweet, isang place
Me,
lang
ma'am.
talaga yung...
Me, ma'am. We do have house sa Conel po. Meron ako sa Conel ngayon, ma'am.
So meron talaga kayong tanim doon ng key lime and
Sa
guava.
ano lang, ano rin, ma'am, sa cousin ko may farm po.
Plantation talaga yun. Isang puno lang yun. Kasi I think baka marami yung kakailanganin ninyo.
Yes, ma'am. Yun rin yung gi-consider ko, ma'am, na hindi lang yung single na isang plant na tinubo na plant kay baka dami rin masyadong leaves ang kailangan for the extraction. So yun, ma'am, nag-choose ako ng area na abundant with that plant.
Okay. See to eat lang din na isang place. Dun lang sa Cornell Creek. With your sampling technique pala, may I add in how to identify young? Kasi na-mention na kanina yung young. Yung sinasabi natin talbos, sa dulo ng guava, makikita mo yung color. So sa color pa lang, makikita mo na kung young leaves siya. Kasi di ba para siyang light green, yung sa pinakadulo. If this is the stem, yung sa pinakadulo niya, medyo light green siya. Yan tama kapag pick mong ganyan, yung stalk niya is easier to cut it out kaysa dun sa medyo old na pag pick mo ganun, medyo mahirapan kayo. Pero pag young leaves pa siya, yung dun sa dulo niya, pag ganun sa stalk, madali siya matanggal. Ayun. So that's the young leaves. Talbos. Okay. Okay na tayo sa research instrument. Yan. Lahat to kayang i-provide ng school. Na-recheck nyo ba? May blender naman din tayo. Working pa yung blender sa school. Check.
Yes, ma'am.
Mechanical mixer. So what we mean by this? Meron ba tayo sa school? Mechanical mixer in formulating their soap? Or iba rin to sa blender, di ba? Ayun lang siguro. Mechanical mixer. So we need to check kung meron sa school. No? Ito.
At the same time, ma'am, yung process of air drying, kasi that has been an issue before. Ma'am Reza,
Yes.
with the recommendation of the panel before, especially with air drying ng mga leaves, it should be really considered kung saan nila ilalagay, na hindi siya ma-expose to dust and other particles. So,
Can they have it sa ilang balay na lang din? Wala mo gutay hangiran sa school. They have to allot a room.
okay lang po ba mga panelists? Kasi that has been an issue before.
Tama. Itong grupo nila ay
Oo, naalala mo yung air drying.
Yes. Wala kasi sabitan.
Oo. Ma'am Mabe and Ma'am Eroy. May naisip yung internet connection nila doon.
Napilit sila din.
Baka mahirap yung
Or kusog man, wala gid ay
nawala. Nawala sila.
Kung mag-air dry sila sa school din, I think we need a room for that. Pero murag gamiton manggod kay mag-limited face-to-face, di ba, ang uban nga versus
so far, ma'am, we have several rooms naman available. If kailangan nila mag-air dry dito sa school, may room tayo. We can maximize the third floor. May third floor pa na doon katabi ng, hindi pa na-set up yung isang laboratory doon. Pwede doon, ma'am, pwede magpa-set up and we can ask the madami ba itong studies na kailangan ng air dry?
Dalawang groups po din.
At least dito controlled siya kumpara sa bahay.
Tapos, pwede lang ta magpabuhat og murag halayan din kay ihalay mo dapat ang tinuod nilang himuon.
Or ang students magawa, ma'am. Di ba may lang sila niyan? Tapos dito lang ilagay.
Sige po,
kahoy. Paggawa sila ng kahoy tapos yung sa bitana na nylon o ano pa.
I-request na lang po din na gamitin nila yung room.
Yung room na lang, ma'am. Pero yung sampayan, siguro nila sila na lang gagawa. Kaya naman nila yun. Kasi lang sa daming dahon, no? Daming dahon.
Daghan na sila gihalay nga dahon.
Pero kung sa bahay nila, kasi depende kasi kung may there are some students na malaki yung area nila. Na may mga extra space sila doon.
Pwede na siguro sa school na lang din para ma-supervise ko din. Di ko kasi makita.
Yeah, para matingnan mo kung paano. Yun nga mas better dito sa school. Dali na lang nila dito, tapos dito na lang nila isampay. I-air dry.
Din brown out daw po sa kanila, Ma'am Eroy. At saka Ma'am Mabe.
Kung burnout, so mahirap ito. Hindi sila mag-reschedule na lang talaga dito, ma'am eh.
Naroon si Ma'am Mabe. Pero si Ma'am Eroy.
So, patapos naman tayo dito sa group nila,
Opo.
Pauline.
So as is po ito din kasi nagbasa po ako
Oo.
ng yung sa recommendation sana ni Ma'am Eroy din na moisturizing activity.
Yeah.
Tinetest siya sa human.
Oo.
Pero mahirap kasi sa human kung student study pa lang ito.
Oo.
And then ang deodorizing activity, ang tester nila is gas chromatography. Ang alam ko lang na may gas chrom din ang UIC po.
At saka hirap at this time, daming restrictions tayo.
Opo. Okay lang po sana kung walang pandemic, kaya po nila.
Oo, lalo na ngayon may Delta variant eh. Even sa NCR ngayon talagang they're making some measures na ngayon na magkakaroon ng 2 weeks lockdown na naman ulit doon sa NCR dito na Delta variant. So we need really also to consider that, no?
Yes.
Hindi natin alam that during the time when they will be conducting the study kung okay na yung natin.
Yes po. So,
situation.
pwede po as is na lang po muna yung study nila din.
Yes, ma'am. For me, di ba, Ma'am Abe? Para sa akin, okay na yon.
Din, brownout po sa amin dito ni Ma'am Eroy.
Yes, ma'am.
I'm using actually dito sa cellphone. Pero si Ma'am magpa-load pa daw. Magpa-load pa siya sa data para makabalik. Nag-brown out.
Okay na siguro ito, ma'am.
After ulan brown out.
Oo, unless may mga iba pang mga concerns. May concern ako sa mga appendices ninyo, no? Like for example, you have written here through this number pero walang number. So if you don't want to write your number here, i-omit nyo na lang. Pa-omit na lang, no? Kasi i-XX nila. You can contact me in this 09XXXX. Kasi if you wanted, no, yung gisulatan ninyo to contact you through email, so yung email address yun na lang. So huwag yung lagyan doon ng XXX, no? Kasi makalimutan nila yan, Ma'am Raiza, then yung isi-send nila sa mga, sa mga offices. Okay? So baka ma-miss out lang. Okay, Ma'am Mabe, do you have any additional recommendations?
Dun sa appendix din is padagdag ng about sa plant. Yung niyo, classification ng plant, parang walang doon. Hindi nadagdag sa appendix nila din. Yun lang. Okay. Paano yan din? Tayong dalawa na lang wala pa si Ma'am Eroy.
I think, na lang, Ma'am Raiza, I don't know, Ma'am Abe, kaya pa ng cellphone mo yung next, natin, yung mga next, presentations. Ang problem natin yan, even students who are situated now doon sa mga areas, baka may kaparehong feather, wala si,
Oo, feather, the same, siguro din.
Buti na ito silang,
Napasilatanan.
andyan sila lahat, walang nawala.
Choppy po ka, Ma'am Mabe, marag hinay imong net.
Oo, sa dito na to, dito.
Napilit na
Dito na yan
yung
ginagamit mo.
Dito din, opo, pang backup. Pero napilit na. I think ang dito kasi okay lang pag sa downtown area. Pero pag medyo remote na, hindi na rin siya ganun kalakas.
Oo, kasi depende, ma'am, kung merong site diyan, cell site din sa'yo, na mas malapit yung site.
Pero narinig ko kayo ng maayos. Baka ako lang, no, ang hindi.
Hindi, okay lang. Audible ka din naman, ma'am.
Mapilit lang siya.
Opo. Okay. So, pwede naman siguro, Ma'am Raiza, we'll just be communicating na lang the result of the defense to the students kasi we need to convene together with Ma'am Eroy in relation to their grade for this afternoon.
So, sa sunod na lang ilahang grade din.
Yes, okay. Yung grade nila to follow na lang. But of course, the proposal already has been done. So it is being accepted. The proponents already know that it has been accepted by the panel. Okay? So, yes.